Welcome to The Explainer. Today we're diving into a story about how one single scientific paper, published pretty quietly back in 1975, ended up creating a $250 billion industry and completely revolutionizing medicine as we know it.
It all kicks off right here. On August 7th, 1975, the journal Nature publishes a paper from two biochemists, Georges Köhler and César Milstein. They'd figured out a new way to create something called monoclonal antibodies. Now, it was a massive scientific breakthrough for sure, but back then, nobody, and I mean nobody, could have predicted the shockwave that was coming.
How big a shockwave, you ask? Well, try this on for size. A global market valued at $250 billion today. And that entire massive industry, which is actually projected to double in the next 5 years, grew directly from the technique described in that one single paper.
So what are these things? Well, today, these lab-made antibodies are the absolute workhorses of modern medicine. The best way to think of them is like tiny, programmable, guided missiles. Seriously. They can be designed to hunt down and attack a very specific target, anything from a cancer cell to a virus, and they're used in treatments that have helped millions of people with everything from autoimmune diseases to allergies.
Section 1. The Great Antibody Puzzle. So, yeah, how did we get here? To understand that, we've got to rewind the clock a bit, because it all started with a huge scientific mystery that had really stumped researchers for decades.
So, back in the 1960s, scientists knew our immune systems could crank out this just seemingly infinite variety of antibodies to fight off all sorts of invaders, but they had no clue how. César Milstein, who was working at the UK's legendary Medical Research Council lab, was a key figure trying to crack this code. The big roadblock, though, was actually studying them. You needed to grow these specific antibody-making cells in a lab, and that was proving to be just unbelievably difficult.
And that brings us to the core problem, the big question. Imagine you find that 1 in a billion cell, the one that makes the perfect antibody against, say, a specific type of cancer. Great. Now what? How do you actually grab that single cell and turn it into a factory that can just pump out that antibody endlessly? That was the hurdle, the wall everyone kept hitting.
Section 2, the hybridoma breakthrough. And the solution they came up with, it was just so elegant. A true masterstroke of collaboration. They called their creation a hybridoma, and the genius of it was fusing two totally different cells to make one supercell. So they took the cell that made the perfect antibody, but, it would die off pretty quick. Then they fused it with a cancerous myeloma cell, which is basically immortal. And what you get is a single hybrid cell that's a perpetual antibody factory. It just keeps going and going.
The process itself was just brilliant. Okay, first you immunize a mouse, right? Get it to produce the antibody you're after. Next, you isolate that one specific B cell from its spleen. Then comes the magic trick. You fuse that cell with an immortal myeloma cell. This new hybridoma had the best of both worlds. It produced the perfect antibody, and it would multiply forever, just churning out limitless copies.
Section 3. An open source revolution. So, they have this absolutely world-changing technology. Now, in today's world, you know exactly what comes next, right? Patents, lawyers, licensing deals, secrecy. But that's not what happened. Not at all.
Instead of locking it down, Milstein's lab treated the discovery like a scientific relay race. They basically saw their job as just passing the baton. So when researchers from all over the world started asking for their hybridoma cell lines, they just sent them. Often for free or just the cost of shipping. You really have to put yourself back in that time. I mean, the biotech industry as we know it basically didn't exist. Patenting research that was funded by the government just wasn't really a thing yet. For Milstein and his team, the goal wasn't to make a product, it was to answer a deep, fundamental question about how our bodies work. And that open, sharing attitude, it let the technology just spread like wildfire, enabling labs everywhere to jump in, test it, and build upon.
Section 4, a lesson for today. In this whole backstory, it gives us this fascinating way to look at how science works today. Because it's a pretty stark contrast.
Think about it. The environment that gave us this breakthrough was all about tackling huge, fundamental questions. The funding was pretty hands-off, and there was a free exchange of ideas and materials. Now compare that to the landscape today.
So often it's dominated by a laser focus on intellectual property, on patents, really complex management structures, and funding that's tied to very specific short-term goals.
The takeaway here is it's undeniable. That free exchange of knowledge, it paid off on a scale that I don't think anyone could have possibly imagined. That one decision, to share instead of to hoard, is precisely what allowed this discovery to create the life-saving, multi-billion-dollar industry we have today.
And that leaves us with a really provocative final thought. In our modern age of patents, of fierce competition over intellectual property, and superstructured research funding, could a discovery this big, this foundational, ever happen the same way again? Could it be born and shared so freely? It's a powerful question to chew on as we think about the future of science and innovation.